want to talk briefly about the different tests available for the SARS-CoV-2 infection, but first I need to define some terms, and we start by talking about comparing whether you actually are infected versus what the test result tells you. So if you are actually infected and you get a test and you have a positive result, we call that a true positive, where a true negative is when you don't have the infection and the test result agrees with that. But we also have something called a false negative and a false positive. A false negative is if you are actually infected, but the test result says you're not. And a false positive is when you are not infected, but the test result tells you that you are. And so the terms we use to talk about these are specificity. So specificity is telling us really the rate of the true positive to the false positive. So we want a high specificity. So that means a high specificity means that we are very specific. So we are specific to the SARS-CoV-2 infection, not any other infection. The other term is sensitivity, and I'm putting this in this false negative box because really it's comparing the false negative to the positive and the negative. So sensitivity is how sensitive we can get. So how few particles can we actually sense? So it's kind of, we want that true positive, and so it's relating all of those negatives to the true positive. So since we can't use real patients to test for these, we use controls. So we have a positive control that is something that we know should give a positive test result, and we have negative controls where we have something that is not the infection that should give a negative control. And so scientists design these to make sure the tests are specific and sensitive. We also have internal controls so that we can compare different things. That's just something that's doped in to each sample that should give a consistent reading regardless of what's happening, so that we can compare everything. So there are 3 tests, at least as is available in the middle of June of 2020. 3 types of tests. The first one is the RNA test. The RNA test, or otherwise known as the molecular test, probes for an active infection. The virus is actually there, and this is detecting the RNA. So to detect the RNA, we must have enough of it. And the thing with this RNA test is it's only positive while you are sick. So the infection's actually there, the virus is present. But it goes back to negative once the RNA is degraded. So once the virus has been cleared from your body, the RNA gets degraded and you would no longer be positive. This has a high specificity, meaning there are few false positives, and it has a fairly high sensitivity as well. We use a method called RT-PCR, and this method works by first extracting the RNA from your cells. So the coronavirus has this RNA. It infects your cells and makes your cells make that RNA. So as long as your cells are infected with the virus, they contain the viral RNA that we extract. So we extract the RNA. The RNA then gets copied to DNA. DNA is much more stable, and it's double-stranded. We copy to DNA, and then we amplify that DNA, and that just means we make many copies of the DNA. So we make lots of copies, and actually while the DNA is copying, we can detect the level of that DNA. And so we do this in an instrument called a quantitative PCR, where we can detect the amount of DNA, and once it crosses a certain threshold, again established by controls, we know that we have a positive result. So this RT-PCR comes from these 2 copying and amplifying steps. The copying is RT. This is reverse transcription. It's the reverse of transcription. Transcription makes RNA. Here we are making DNA. And then PCR stands for polymerase chain reaction. This is an enzymatic reaction that faithfully copies the double-stranded DNA. So there are different versions of the RNA test, of this molecular test, and they differ mostly at these 2 points, the extraction and amplification steps. The extraction step, there are methods that can do this more rapidly. The sensitivity of that, so how low it can sense, is questioned. More tests are being done to confirm that sensitivity. And then the amplification often needs to be done on a specific instrument, but they are making more tests available that can be done more rapidly and at a point of care, as a point of care diagnostic. So again, this is improving, but the basic principle is the same. We extract RNA, copy it, and amplify it, and then use some sort of detection method. The next type of test is the antigen test that also probes the active infection. This is the newest test to hit the market. So again, there's a lot more testing of the test that needs to be done. So when we say it probes the active infection, the virus is still there. This is detecting the proteins instead of the RNA. So how this works is we first release the proteins from the infected cells. And if you remember, we have lots of proteins in the virus. We have those spike proteins, that's pink shown in this figure. The green and blue are also different proteins. But there's a protein that actually coats the RNA. And this is the protein that's been most successful in the analysis. So I've made that yellow here, but we get all of the proteins out. So we have all of the proteins. Then we flow this protein extraction over a strip that contains antibodies. Antibodies we often draw as these Y shapes. It's similar to how the structure actually is. And when we flow the proteins over, if the proteins are present, they bind the antibody and we can detect that. So while this is a newer test, it's thought that it actually has a lower specificity. So these proteins are a lot more similar to the original SARS-CoV virus than the RNA is to the original SARS-CoV virus. And so this can also detect the original SARS-CoV virus, but since that is not in circulation, it's thought that this lower specificity might not be that big of a deal. But this test can be a little bit more robust. Proteins are sometimes easier to extract than RNA. And then finally, another test. So both of these antigen and RNA tests are serological tests. So you need a nasal swab or maybe some saliva sample. But the antibody test is a blood test. This detects antibodies. So in response to any foreign object, your body produces antibodies. And it takes a while for those antibodies to be made. So this will not be positive until your body has made enough of these in response to the SARS-CoV-2 virus. And again, these antibodies are found in your blood, so that's why you extract the blood. These are good to detect who has had the virus because antibodies last longer than the virus does. So the sensitivity originally was questioned, but the sensitivity of these has improved. So what I most recently read is that the sensitivity is becoming a lot more robust than it was originally. So we have fewer false negatives. So how this works, again, we have fewer false negatives. How this works, we extract antibodies from the bloodstream. Then we add viral protein pieces. So these are not the actual virus. It's just pieces that look like the virus. We add these together, and if they bind, we get a signal. So while this is helpful to see who has had the virus, two caveats that need to be mentioned is we don't currently know if you can get reinfected. So the hypothesis is that you can't, but that's not necessarily been validated yet. And also we don't know how long antibodies will last. So again, currently the hypothesis is that the antibodies will last a while and that you cannot get reinfected. But more testing needs to be done to prove that hypothesis. So while we can operate under that principle for now, don't be surprised if it changes. Science is continually being improved upon, and so these hypotheses hypotheses could change. So I wanted to share this information about the tests, particularly the RT-PCR test. That's something I did in my graduate studies a lot of, and so I wanted to share a little bit of my knowledge with you. I hope you enjoyed it.